Today we're going to talk about reverse transcription, but before we do, I found an article which is perfectly relevant to what we discussed recently, and it has to do with the structure of vesicular stomatitis virus. You may remember, maybe not, I don't know, maybe you don't remember talking about this virus, but this was an enveloped virus, negative strand RNA, schematic on the left. The RNA, you remember, is complexed with a nucleocapsid protein, and the protein is repeated on the RNA many, many times. We draw it like this. And on the right is the nucleocapsid itself, the RNA, the green strand wrapped with the protein. And a paper came out last week in Science, which is the cryo-EM structure of VSV. So it's Science, February 2010, 5th February. Cryo-EM model of the bullet-shaped vesicular stomatitis virus. So from what you've learned so far, you could almost understand all of this paper. VSV is a bullet-shaped rhabdovirus in a model system. Through direct visualization by cryo-EM, we show that each virion contains two nested left-handed helices, an outer helix of M and an inner helix of N in RNA. So that's the interesting part about this. They show that the N protein, which coats the RNA, interacts one-on-one with the M protein, which is underneath the membrane. And this is very unusual. Most envelope viruses have a loose shape, and VSV is one of the exceptions. It has this very rigid bullet. So now the structure is going to explain that. And you may remember some alpha viruses have a rigid shape also, despite being enveloped. They have an icosahedral capsid wrapped in an envelope, but contacts between the glycoproteins in the envelope and the capsid confer the icosahedral symmetry of the capsid to the glycoproteins. Okay, so that's one way you could get an enveloped virus to have symmetry on the exterior. This is another way. So let's take a look at this cryo-EM structure. All right, so these are just some diagrams from the paper. Just to give you an idea, so remember cryo-EM, you take particles, you don't stain them, you just freeze them in water. And they have very low contrast, but it's enough to see the image, so here are the bullets. You take many pictures of different particles, and the assumption is that each one is in a slightly different orientation. And then you use computational methods to calculate a model. So here's the cryo-EM model of the virion. The envelope is in two different colors. The outer leaflet is in magenta, and the inner leaflet is in dark blue. Then underneath it we have the M protein, which is in blue, and then the N protein is beneath it. So this would be complex with the RNA. But we don't see the RNA in this structure. Do you know why we don't see RNA in this cryo-EM structure? What's the resolution of cryo-EM? Does anybody remember? Is it 1 angstrom? Is it 10? Yeah, it's 10 angstroms. It's not great. And the diameter of RNA is about 2 angstroms, so you can't see it. It's just not high resolution. All right, so the N would be bound to the RNA. But the key here is that, look, the M is following the helix of the N protein. It's very interesting. That's what's new about this. Okay, so let's see a movie of this. They're rotating it so we can see, and they've cut away part of it. They've also cut the top away, which is the bullet part, of course, which we'll get back to. This is very interesting. So here's the N protein making its nice helix, right, winding down, and the M protein contacts it. But it's very interesting. So the M makes a helix as well because of the N protein. It's very exciting, isn't it? A lot of action. So once again, the outer leaflet, the inner leaflet, M protein, N protein. So this explains why the capsid is very rigid. Of course, they've taken the top and the back off as well, so you can see it, and they've cut out this bit here. Okay. So what about the bullet top? So it turns out they saw the bullet top in the structure as well. It turns out that the helix of the N protein has to be different to make the cap. You can see that the N subunits near the tip of the virion, or the bullet head, are differently oriented from the N subunits further down. So as you get nearer to the tip of the bullet, there are fewer N proteins per turn, and they're tilted so that they are in a different orientation compared with the rest of the nucleocapsid. And their model is that this is what happens first, that the top of the bullet forms and then the nucleocapsid wraps around it with the RNA in it, which is shown here. And then when it gets to, I think, the 7th layer, it becomes in the correct, this is the first helical layer, as it is in the rest of the molecule. So that explains how the top is made. You may remember I showed previously a structure of N protein complexed with a very short RNA, a few subunits of N protein plus RNA, and that structure was solved by high-resolution X-ray crystallography. They were able to fit that structure into the N protein structure determined here by cryo-electron microscopy, showing that that structure is correct. Also in that structure, they knew where the 5' and the 3' end of the RNA is. So from that, they could tell that the 5' end of the RNA starts up here, and the 3' end is down at the back, which is something that had been assumed before, but had never been proven. All right, then the final thing, which is very interesting here, is this detail up close. So here is the outer leaflet of the membrane and the inner leaflet, and this is a single M linked to a single N protein. Right? If you just take away everything else, you can zoom in. And then we're zooming in on that complex here. And look at this. There's a little bridge between the M protein and the membrane. And they suggest that this is the tail of the glycoprotein that's coming through the membrane. So remember, on the outside of this membrane, which is not shown, are all the viral glycoproteins. And they pass through the membrane, and they usually have a little inner sequence, and they think this is this interacting with M protein. So that would give the outside of the virus order as well. And if you look at negatively stained EMs, you see that the glycoproteins are ordered. And this is probably why that's so. So probably this whole thing is made very rigid by all these interactions from the glycoproteins to the M and to the N protein. All right, today we're going to talk about reverse transcription and integration, and a little bit of history to put this in perspective. In 1908, the first tumor virus was discovered by Bang and Ellerman. This was a virus that caused leukemia in chickens. But nobody really cared about this, because they believed that cancer in animals was totally different from that in people. 3 years later, Peyton Rouse, who was working at the Rockefeller Institute in New York, was brought a chicken by a farmer from New Jersey, and it had a big tumor on it. And he took this tumor and he ground it up, he filtered it, and he injected the filtrate into new chickens, and they got tumors. By passing the tumor filtrate through a small filter that held back bacteria in cells, he showed that it was a virus which, when injected into a new chicken, caused the formation of a solid tumor or sarcoma. So two very close discoveries on tumor-forming viruses. Remember, by 1908, it had already been established that viruses existed, that they were very small organisms that could pass through a filter. These were really the first cancer-causing viruses to be discovered, and at this point they were simply called tumor viruses. But nobody cared about this stuff. It was amazing, and he got a Nobel Prize 55 years after he made this discovery. This is the longest gap between a discovery and a Nobel Prize ever. And finally people began to realize that this was important stuff. So Rouse sarcoma virus. By the 1950s and 1960s, these tumor viruses were shown to have a genome of ribonucleic acid, or RNA. People began asking, how could these RNA viruses transform cells? We know if it were a DNA virus, that DNA could integrate into the host and transform it, but these are RNA viruses. What is going on here?
such that the virus infects the cell and for the life of the cell it's transformed. It's oncogenically transformed. It can cause tumors. Well,
two people thought about this problem a lot, Howard Temin and David Baltimore.
And they discovered the enzyme that does this in 1970. It was called reverse transcriptase, and they got the prize 5 years later, discovery. Reverse transcriptase.
And it was called that because it was the enzyme that countered the central dogma. Instead of DNA making RNA, which then made protein, this enzyme took RNA, made DNA out of it, which eventually went on to make RNA again and protein. The viruses we're going to talk about today are here group 6 in the Baltimore scheme, and the same scientist who put together this classification according to genome types also was one of the discoverers of reverse transcriptase. Howard Temin had the idea before the discovery of reverse transcriptase that somehow these viruses integrated their genomes into that of the host cell, and that was how they caused transformation, oncogenic transformation of the host cell. Temin hypothesized that in some way these viruses converted their RNA genome into a DNA copy, which was then integrated into the host chromosome. And for many years he tried to find evidence for retroviral DNA, or at the time tumor viral DNA, in host cell genomes, and he couldn't find it. The techniques were simply not available back then to make that possible. Today we could easily detect integrated retroviral DNA in a host cell using, for example, polymerase chain reaction, but these techniques were not available when Temin was working on this problem. At about the same time, both Temin and Baltimore had the idea that maybe there was an enzyme in the tumor virus virion that converted RNA to DNA, and they both started looking for that enzyme. Now remember from our talk about RNA synthesis, Baltimore was one of the first to discover an RNA-dependent RNA polymerase in cells infected with plus-strand viruses, and then in the virions of negative-strand RNA viruses. So he was used to the techniques for discovering these enzymatic activities, and he reasoned perhaps there is an enzyme that converts RNA to DNA in these RNA tumor virus particles, and I'll simply use the same technology to detect it.
But it's not intuitive, right? These are plus-strand viruses. So why is it not intuitive that they said there would be a polymerase in there?
Does anyone recall why it's not intuitive? Because if it's a positive strand RNA virus, you can translate the genome as the first step coming into a cell, and you don't need a polymerase in the particle.
And that's what I've told you many times, that if it's plus-stranded, it just needs to get in the cell and be translated. And I always say, but the exception is the retrovirus. So why would they do that? Well,
it was a long shot.
That's the only answer we have, but it worked.
That the plus-stranded RNA viruses do not need an enzyme in the virion. They did not work in a vacuum.
Since the early 60s, many labs have been studying bacteriophage lambda, and it had been
shown purely by genetics
that the DNA genome of that virus can integrate into the bacterial genome.
The bacteriophage lambda has a double-stranded DNA genome,
shown here, and it was known that this could circularize
when it infects a cell, and then this circle could integrate into the bacterial chromosome at a very specific site,
the at-B site in the bacterial chromosome, and then you would have an integrated copy of lambda DNA, which is called a prophage. So this had all been sorted out genetically. Nobody sequenced back then, but Alan Campbell and many others deduced from genetic crosses that this was so, that the genes were in different organizations in circles versus integrated prophages. And now many years later, of course, we know that this is true.
So Temin got the idea for retroviral DNA integration from bacteriophage lambda, and he called the integrated retroviral DNA a prophage, a provirus, rather than a prophage.
So these are the viruses we're going to talk about today.
There are many different kinds of retroviruses,
but I just want you to be concerned with the concepts that are common to all of them. Here are the particles by electron microscopy. They are enveloped and they have a core. You can see a dark core here. For many of these viruses, that core has icosahedral symmetry.
So you know how that's built.
So here's the diagram on the right. There's an envelope which is studded with glycoproteins.
The envelope glycoprotein is made up of two subunits. One is called SU for surface. The other is called TM for transmembrane. Just below the lipid membrane is the matrix protein, the MA, which in other viruses is simply called M, and this gives the envelope some rigidity. And then below that is the nucleocapsid, which in many of these viruses has icosahedral symmetry. Within the nucleocapsid are the viral RNAs, and as we'll see in a moment, there are two plus-stranded RNAs incorporated into each particle. There are also several different kinds of proteins within the nucleocapsid core. There's the reverse transcriptase, there's a protease, and an integrase, which we'll talk about shortly.
Now the genome is laid out in this way, and I just show you this to give you some sense for how it's expressed. There are simple and complex retroviruses. Complex retroviruses are like HIV. They have many more proteins encoded in the genome. But for now, we're not really concerned with those. So we'll look at a simple retroviral genome. This is the proviral DNA. So this is a map of the DNA that would be integrated into the chromosome. And we'll find out exactly how that works in a bit. And these are the three general areas that encode different gene products. So first of all, you have at either end of the proviral DNA, you have this sequence called the LTR, the long terminal repeat, and we'll talk about what that means in a moment. And then this genome encodes three general classes: the core, or the proteins that make up the capsid, and that's called the GAG region, for group antigen, which is an old name that has stuck, derived from immunology. And among these proteins are the capsid protein,
the main protein that makes up the icosahedral shell. Then we have enzymes. In the Pol region, we have reverse transcriptase,
which actually has two activities, we'll see, and the integrase, which is responsible for the integration of the DNA product. And then we have the envelope proteins, the surface and the TM, which form the glycoprotein, and that's called OMV.
These are all expressed from messenger RNAs made from the integrated DNA copy of the viral genome.
So you have a large RNA shown here. This is called genomic RNA because eventually this is what's packaged into new virions. When this is translated, you mostly get the GAG precursor, which gives rise to all the structural proteins by processing. There's a termination codon here, and a small fraction of the time the terminator is suppressed and you get the synthesis of a GAG-Pol fusion protein, and that allows you to make the enzymes which you need, so they're essential. So you don't get as much of that, but they're enzymatic, so you don't need as much as a structural protein. So among those are the reverse transcriptase and the integrase.
To obtain synthesis of the envelope proteins requires splicing of the genomic RNA to remove this sequence so that the envelope coding region can be translated. Let's take an overview of the early stages of a retroviral infectious cycle and then look at some of the steps in detail. As all the other viruses we've talked about so far, these virions bind host cell receptors and are taken up into the cell. Some of the virions fuse at the cell surface, others are taken up by endocytosis. The virion is partially disassembled in order for the viral RNA to be converted to DNA by reverse transcriptase. But the capsid is never completely disassembled.
So those RNAs are never floating free in the cytoplasm. They stay within a structure, and within that the viral DNA is made. So the genomes are converted to a double-stranded DNA complex. This is called the pre-integration complex, or PIC. This is composed of a plus and minus DNA copy of the RNA. So you go from two RNAs, two plus RNAs, to a double-stranded DNA plus-minus configuration. This gets into the nucleus, and then the double-stranded DNA gets out and integrates into chromosomal DNA,
at which point it's called the proviral DNA. Also in the nucleus, there are formed circular versions of the double-stranded DNA copy, but we won't consider those at this time.
All right, from the integrated proviral DNA, the host RNA polymerase, DNA-dependent RNA polymerase, POL2, makes messenger RNAs that give rise to all the viral proteins, the glycoproteins, the structural proteins, and the viral RNA, which is simply a full-length message produced from this integrated DNA. And that's how new virions are produced, which are composed of two RNAs again, and all the structural proteins, and they're released by budding. So let's look at how some of this works. First, reverse transcriptase, like most of the other polymerases we've talked about, is a primer-dependent enzyme that requires base pairing between the primer and the template, and which produces a new strand in the 5' to 3' direction.
The primer can be DNA or RNA, it doesn't matter which. The primer must have a paired 3' hydroxy terminus.
Primer template components where that last base is not paired, it won't work. It wants to have that last base paired.
The template for the enzyme can be RNA or DNA. It doesn't discriminate. So the incoming viral RNA is converted to DNA by the enzyme, reverse transcriptase, and then that negative strand, the single-stranded DNA, which is of negative polarity, is converted to a double-stranded DNA form by the same reverse transcriptase. Reverse transcriptase will incorporate deoxy-NTPs, not ribo-NTPs. The enzyme makes DNA, not RNA. And lastly, the enzyme selects the correct incoming dNTP by base pairing with the template. The second enzymatic activity of reverse transcriptase, in addition to nucleotide polymerization, is RNase H. This activity cleaves RNA only when it's in a duplex or double-stranded form. The duplex can be in the form of RNA-RNA or RNA-DNA. It doesn't matter. In either case, it's the RNA component that is degraded by the enzyme. The enzyme makes endonucleolytic cleavages. That means it cleaves internal in the RNA sequence as opposed to cleaving at the ends and chewing the ends in, which would be an exonucleolytic activity or an exonuclease. The endonuclease activity produces short oligonucleotides with a 5' phosphate and 3' hydroxyls. The enzyme cuts between the phosphate and the oxygen. Here's the viral genome as it appears in the virion. It consists of two full-length plus-stranded viral RNAs.
These are messenger RNAs. They're capped at the 5' end. They have poly A at the 3' end, but they're not translated when the virus comes in the cell. They could be. You can take these RNAs and put them in vitro reactions. They will be translated, but
as they come into the cell within the virion, they are not translated into protein.
They have some complementarity, which gives them this structure. So you can see there are areas where the two strands are brought together. Slightly offset, there's a little bit of complementarity in different regions which hold the two strands together. This just shows you on these RNAs the GAG, POL, and envelope coding regions, which we talked about. And then the other thing that's present in the virion, besides the RNA and the enzymes, are tRNAs. And these are specific tRNAs depending on the virus, which serve as primers for reverse transcriptase, which we'll see. And the other thing, of course, is that this RNA is coated with nucleocapsid or NC protein. And this is unusual for a plus-stranded RNA virus, right? Because most of them would get in the cell and they would be translated, so they don't need to have NC. NC usually coats an RNA when that RNA has to be replicated in some way when it gets in the cell.
The negative strand RNA genomes of influenza virus, vesicular stomatitis virus, and many others are coated with a nucleocapsid protein
to get the RNA copied by the polymerase in the particle. So this RNA has a polymerase in the particle, and in order for it to work, it has to be coated with NC protein. There are about 50 to 100 molecules of reverse transcriptase per virion.
The fact that there are 2 copies of the genomic RNA in each retrovirion is very interesting. It's been known for many years that retroviruses are relatively resistant to ultraviolet light and ionizing radiation.
More so than other viruses of the same size genomes, and we think that's because there are 2 copies of all the genes.
The polymerase can, for example, use undamaged parts of either RNA to make up a complete infectious genome.
So if there are mutations or breaks or deletions in one, the enzyme could use the other strand to correct it.
That's why it's believed that there are 2
RNAs in the genome to give the virus the ability to correct mistakes.
How the retroviral reverse transcriptase knows when it encounters a mutation or a deletion in the genome isn't known, nor is it known why this strategy isn't followed by other viruses with plus-strand genomes. Those viruses have single copies of their genomes in virions, but it must have some evolutionary advantage for this virus to use this strategy.
The first thing we're going to look at is the way retroviral reverse transcriptase primes DNA on the template. So here again is the viral RNA. We're just looking at the very 5' end. This is a short bit of the 5' end. There are very specific sequences here, and you'll see them again, and I want to familiarize you. One of them is called U5 because it's in the 5' untranslated region of the mRNA. And this is composed of a variety of inverted repeats, which we won't worry about. But then just downstream of the U5 region is what is called the primer binding site, or PBS. And this is the site for the binding of the tRNA, which is
the primer for reverse transcription, for the initiation of viral DNA synthesis. Now on the top you can see a schematic of the tRNA bound to the 5' end of the viral RNA. This, of course, is a cellular tRNA, so the virus is using a cellular RNA to prime DNA synthesis. On the lower left is the structure of the primer binding site in the absence of the tRNA. You can see that it's a highly structured RNA. It forms a very tight stem loop. Next to that, in the middle, is the structure of the tRNA. And then on the right is the structure of the tRNA hybridized to the primer binding site on the viral RNA. You can see that the tRNA has melted the middle of the stem of the RNA, and it's insinuated itself into that stem by hybridizing to the bases. By doing so, the 3' end is now in a position to act as a primer for viral DNA synthesis.
So let's go through the steps of DNA synthesis.
On the left is the viral RNA in Kelly green with the tRNA primer hybridized near the 5' end.
The reverse transcriptase uses it as a primer to start making DNA. So this is light blue because it's negative strand DNA. This primer is right near the 5' end of the RNA, so not very much DNA is going to be made. It's a very short piece.
It's actually called minus strong stop DNA because in the early days when it was first discovered, it was discovered as a small fragment on a gel, and people said, what's going on here? There's a small piece of DNA. There must be a stop in the viral RNA which is causing this. So they called it strong stop DNA.
But it turned out it was reaching the end of the template.
As this short negative strand DNA is made,
the RNase H activity gets rid of the RNA that templated it. It's not needed anymore. And it wants to get rid of it because this is now going to hybridize with the sequence at the other end of the RNA. So the two ends have a complementary sequence, and this is now going to serve as a primer here. So there's nowhere to go here, of course, so what it's going to do, it has to get to the other end of the molecule and start copying.
Here's the next step. It's called the first template exchange, because the short negative strand DNA that has been made reaches the end of the RNA template and then begins to prime at the other end of the viral RNA, at the 3' end.
And the enzyme continues, and it continues to make negative strand DNA, which is the light blue, and as it does so, it chews away the template as it goes along. You can see here getting all the way around the genome until it gets near the end. But you notice that it leaves one piece of RNA. It doesn't chew it up. This is called PPT, or polypurine tract. It is the enzyme, yes, we'll use that as a primer to start making plus-stranded DNA, which you see is the dark blue DNA right here. So as this is happening, the PPT is priming plus-strand DNA. All right, and then finally the negative strand is completed. We're all the way back to the primer binding site. At the same time, the PPT primed plus strand is here, and it reaches the tRNA. So something has to happen to complete the genome, and what happens is the tRNA primer is removed from here, and this new plus strand can anneal with its complement on the negative strand and starts to elongate there. So this tRNA sequence is actually copied twice, as you can see here, once from the genome and once from the primer. And so here we are now with this molecule. You have an almost full-length minus strand with a little piece of plus DNA. And this is simply going to be elongated. Go down to the left here. This will elongate all the way around and make a final double-stranded copy of the genome. Don't worry about any of this on the right here. All right, so it's a full-length double-stranded DNA, and what is interesting has happened is that the ends are now repeated. By this funny jumping and copying of ends, we've got now the U3R-U5 sequence here, and it's repeated, U3R-U5. Okay, so the messenger RNA didn't have that at both ends. The messenger RNA started about right here at the U5 and ended about in the U3. So this has got extra sequence, and it needs the extra sequence in order to make messenger RNA, as you'll see, and integrate into the host. Okay, so that's how the RT works from a strand of RNA, two strands of RNA. It makes one double-stranded DNA copy. Okay, everybody okay with that? Yes?
Are there any stranded retroviruses?
No, there aren't any negative stranded retroviruses that we've discovered yet. No. Well, theoretically, no, right? A negative stranded genome could have a reverse transcriptase in it and would copy it. No theoretical reason, no.
There are DNA viruses that encode reverse transcriptase, but no known negative strand RNA viruses with reverse transcriptase. This slide illustrates the structures of reverse transcriptases from various retroviruses. So we have three different retroviruses here. ASLV is avian sarcoma and leukosis viruses, MLV, murine leukemia virus, and HIV-1. And for the ASLV viruses, for example, the reverse transcriptase is a heterodimer of two proteins. At the top of this slide, by the way, is the mRNA encoding the Pol region, and below it are the protein products. So the ASLV reverse transcriptase is a heterodimer. One polypeptide consists of polymerase, RNase H, and integrase as a fusion protein, and the other subunit consists of polymerase and RNase H only. A cleavage shown by the open triangle has occurred which removes the integrase from that polypeptide.
So this open triangle means it's cleaved some of the time here by the protease, the viral protease. The murine leukemia virus is just polymerase and RNase H.
Integrase is a separate subunit. It's been cleaved from the RT RNase H polypeptide, and this happens all the time as shown by the solid red triangle.
And then the HIV can have both in the virion, both a fusion of polymerase and RNase H and polymerase alone, and again, integrase is separate. We don't know why this happens, but they're all shoved together in the virus particle, and they're all close to each other, these subunits. Now remember the sequence of relationships that we talked about when we talked about RNA-dependent RNA polymerases. There are 4 classes of
nucleic acid polymerase,
and they all have sequences in common. And here's reverse transcriptase here underneath RNA-dependent RNA polymerase, and they have these regions of sequence homology. There are 5 between these RNA polymerases, and these are reflected in the structure of the proteins. It looks like a right hand, just like all the other polymerases. So here's this
X-ray structure of reverse transcriptase. At the top is the alpha carbon trace, that is the amino acid backbone of HIV reverse transcriptase, with the different domains in colors. You can see the palm, the fingers domain colored here, and this is a structure with the primer and template strand as part of the enzyme. So the template strand is blue and the primer strand is light blue, and the incoming deoxy-NTP you can see on the left is the next one that's about to be polymerized. At the bottom is a cartoon of the RT structure. It's colored according to domain, so the fingers on the left are in yellow, the thumb is orange, and the RNase H is magenta. You can see that the template strand, which is the RNA, the plus strand RNA, is coming in at the left. The first site is the polymerase active site, where the deoxy and NTPs are added. There are 2 white circles there, which are where the metal cations are bound, and these are the metals that are needed for catalysis. Then you see the production of the blue product, the negative strand DNA, and that proceeds to the right. And as that product template passes through the RNase H domain, which is in magenta, the RNA strand is degraded so that what comes out of the enzyme on the right side is just a single-stranded DNA product. The RNase H also requires divalent metal ions for activity, and those are also shown as circles in the diagram.
And this is a very slow process. It's about 10 times slower than other enzymes that we've talked about. It takes about 4 hours to make a DNA copy of one retroviral genome,
which is about 9 kilobases in length.
4 hours. Very slow. It's also error-prone. 1 misincorporation per 10 to the 4th to 10 to the 6th nucleotides, depending on the conditions in the viral polymerase.
If you're infected with HIV-1 and you are taking antiviral drugs, you develop resistance to those drugs because the polymerase makes errors, some of which encode resistance to the antivirals. So this enzyme is making errors going from RNA to DNA.
Whereas in the RNA viruses, it's from RNA to RNA. Because the polymerases are error-prone, there's no error correction, either for RNA-dependent RNA or RNA-dependent DNA polymerases. So far, we've made a double-stranded DNA copy of the genome. This happens largely in the cytoplasm. The whole complex goes into the nucleus. It's called, again, the pre-integration complex. And now we're going to look at this step of proviral integration. Well, how does that happen? So here's the linear DNA that we've just made. Remember, it's a full-length copy of the mRNA of the virus, plus it has an LTR now at both ends. What's going to happen is this is going to integrate into a host target, such that now we have an integrated viral DNA, the provirus. It's going to have an LTR on both sides. It's going to have lost the 2 terminal nucleotides of the viral DNA. Okay, so the AA here, TT here is going to be lost.
And in addition, the host target sequence is going to be duplicated.
So you see there's a little red host target here. You now see it at either side of the proviral DNA. When people look at viral DNAs integrated into our chromosomes, you can tell if it's a retrovirus because of those 2 features, duplication host DNA and loss of the viral sequence at the ends. So there are many other ways that DNA can get into the genome, but if you see those two features, you can suspect
that it's a retrovirus that has integrated from those features. Once the proviral DNA is integrated into chromosomal DNA, that is when messenger RNAs are produced from which the viral proteins are translated. And you can see here the viral RNA genome is produced by transcription of the proviral DNA. It begins within the LTR, and it's mediated by host DNA-dependent RNA polymerase II. How does integration of the proviral DNA occur? That's mediated by the viral enzyme integrase, and it occurs in 3 steps as shown here: processing, joining, and repair.
So this little bone-like structure outlined here on the left is the integrase protein, and here's retroviral DNA. So it's the full-length, double-stranded retroviral DNA as the reverse transcriptase has just made it. See, everything's intact. First step, processing. A divalent cation-dependent reaction. The 2 nucleotides at each end are taken off at the 3' end. So the 2 terminal 3' end bases are removed. And the second step, these free 3' ends invade the host DNA or the target DNA, which is shown in purple here. So each 3' end is now going to invade this phosphodiester bond, which is shown by a little yellow circle. So these break, the 3' ends break the bond in the host DNA, and then they are joined to it. And in fact, the energy released by breaking this phosphodiester bond goes towards making the bond between the viral DNA and the host DNA. So you have the structure shown on the upper right. So now we have 1 strand of each joined. And there's a free 5' end here, so that has to be fixed. That is repaired. It's filled in, and now we have a double-stranded region here completely, and you see why you have duplication of the host sequence, because you're filling in the same strand, the same region, just 2 different strands of it. So this and this are the same sequence, and when you fill them in, you get a duplication. So you process, you join the ends, you repair by filling in the extra nucleotides from whatever has been chewed out, in this case 2. And now you have an integrated copy of the viral DNA, which is called a provirus.
The repair that occurs after integration is carried out by host DNA repair enzymes. This is integrase, the enzyme that carries out the integration reaction. It is encoded in the Pol region. You remember the polymerase encodes reverse transcriptase, RNase H, and finally integrase. At the top is a schematic of the protein, and at the bottom is the alpha carbon trace of the high resolution structure. Integrase is a zinc finger protein, so zinc, shown in the green area, is essential for catalysis. This is a figure from Flint, the textbook for this course, and it's mislabeled. The C-terminus, which is labeled after the zinc binding region, is wrong. That's the N-terminus. And the red region, the DNA binding domain, is the carboxy terminus. So the bar figure at the top of this image is labeled correctly, N-terminus at the left, C-terminus at the right. Whose job is to do the reactions that lead to integration, the cutting of the ends,
ligation to the host DNA, and then we have a DNA binding domain. This enzyme is active as a dimer, shown here, 2 copies of the protein.
We have the catalytic core at the bottom left,
The C-terminal DNA binding domains and the zinc fingers up here, and this is how they align
the 2 DNA substrates of the reaction.
So here's one subunit of integrase, and here's the other subunit. So one is in green and one is in orange.
Here's the target DNA lying across the catalytic region, and these
2 phosphates here circled in yellow background are the ones that are going to be cleaved
by the 3' hydroxyl of the viral DNA. In this image, the 2 3' bases of the viral DNA have been removed, Remember, this DNA end was flush originally before integration began. Now we have a 5' overhang, a recessed 3' hydroxyl, which is going to carry out the attack reaction on the target DNA. As we mentioned before, reverse transcription occurs in the pre-integration complex in the cytoplasm. You can see here that we have the PIC containing double-stranded DNA. Both strands are present. That complex is then imported into the nucleus where integration is going to occur. And a number of cell proteins have been implicated in the integration process, and these are shown on this diagram.
HMG, LAP2-alpha, BAF, Amarin, these are all host proteins that have been implicated in the process. So apparently not just viral proteins are necessary for this integration step. For example, Amarin is a cell protein that is bound to the nuclear envelope. And it's thought to attract one of the proteins in the pre-integration complex because active chromatin is usually in the periphery of the nucleus. So here's some active chromatin here. This is host cell chromosomal DNA wrapped up in nucleosomes. And when it's transcriptionally active, it's usually at the periphery. So it's thought that this interaction between the pre-integration complex and Amarin helps keep the PIC where the active chromatin is. There's another protein called LEGF, which is essential for integration. It's thought to target the integration to exposed parts of the chromatin. So you know the DNA is wrapped around these nucleosomes, and a lot of it is not accessible. But there are some areas that are, and this protein may help the pre-integration complex target those. So where do these viral DNAs integrate? Do they integrate everywhere? Well, they don't. There are preferences. They integrate on any chromosome pretty much, but depending on the virus, there are certain kinds of preferences. So if you look at all the genes, this has been studied in many labs. Thousands of integration sites have been identified and sequenced to try and get an idea of what's going on. And if you just look within genes or at transcription start sites, if you look randomly, say a random integration, this is the ratio you would expect, 26 hits within a gene and 5 within a start site. That's just because there are more genes than, about 5 times more genes than start sites. And then you can see the numbers in human cells for 3 different retroviruses, ASLV, MLV, and HIV. This is percent integration. You can see that ASLV likes to be within genes pretty much, but MLV, really, it's almost 50-50. HIV also prefers to be within genes. So it depends on the retrovirus. Most of the time, they like going within genes, but others also go to transcription start sites. We don't know why that is so. Here is an experiment to see the function of LEGF, that protein which I told you might help target the pre-integration complex to exposed chromatin. You can see in a wild-type mouse cell, HIV prefers to integrate within genes. But if you take away LEGF genetically from the mouse cell, you now reduce the amount of integration in genes and you increase the integration within start sites. So this is part of the evidence that this protein has a role in integration. So once the DNA of the virus is integrated, it's permanently part of the host genome. This is the problem with retroviral infections. You can't get rid of it. The virus doesn't take it out, it just leaves it there. So there's no mechanism for excision. And as a consequence, many proviruses become fossilized. In fact, they've been in us, as you'll see, for 40 million years. They don't leave. And as we say, our genomes are littered with ancient and modern retrotransposons, which are basically elements containing RT. And these are believed to be sources of new genes. The only way for the virus to get out of this is to transcribe, to make RNA, and then it can make new viruses and get out of that cell. But the DNA is always left behind. And that transcription, again, is carried out by host RNA polymerase II. So as soon as the virus is integrated, the host is really driving the production of new virions. So this is very interesting, that there's a promoter built in to the LTR, and then that drives host transcription to make viral RNA, which is then made into protein and particles. There's no DNA replication. There's no RNA replication for these viruses. So far we've talked about RNA viruses that encode enzymes that replicate their genomes. This virus doesn't do that. It has a reverse transcriptase which makes one DNA copy per 2 RNA molecules. So that's not replication, right? It's actually destructive replication because you go from 2 RNAs to a single DNA. And then once the DNA is integrated into the host does everything else. So this virus doesn't actually encode an enzyme to replicate its genome. And all the other viruses we're talking about do that, but not this one. So that's amazing. It's really an interesting strategy, and obviously it works because these viruses are prevalent. So then here we are, proviral DNA, just to end this up. We're integrated, and we're making mRNAs from the host enzymes that are then translated into proteins to make new particles. Now, retroviruses are not the only viruses that have reverse transcriptase.
I want to tell you about 2 other families of viruses that encode reverse transcriptase, but these are DNA viruses. That is, they package DNA in the virion. The first is hepatitis B virus, which, as the name indicates, causes infections of the liver.
And gets a lot of attention because not only do a lot of people globally have hepatitis B infections, but it often leads to hepatocellular carcinoma. So it's a big inducer of cancer.
Specifically liver cancer, and people are trying to find out how it does that. This is the virus with the odd genome that we spoke of earlier. The genome is double-stranded DNA, as shown in the lower left, but it's not completely double-stranded. There is a gap. In addition, there's a protein and a piece of RNA linked to the DNA genome. Today we're going to find out how all this happens. The circular viral DNA is about 3.4 kilobases in length.
And it encodes a variety of proteins, which are shown here on the outside. You can see that a bunch of messenger RNAs are made that encode each of these proteins. The most important messenger RNA is this one, called the pre-genome RNA. It starts here about where this protein is, goes all the way around, is a complete copy of the DNA, and then goes a little bit further from where it started. That's the pre-genome RNA, and
that will be used to produce new DNA viral genomes by reverse transcription.
Okay, so let's see how that happens. So the way these viruses work, they bind to receptors, get in cells. We don't really understand how these viruses get into cells, but the important part is that the DNA genome gets in the nucleus where it's repaired.
The gap is repaired, the protein and the RNA are removed, and the DNA is made covalently closed and circular. It's believed that the repair processes are carried out by host enzymes. In the nucleus, the DNA, the viral DNA, is transcribed by host DNA-dependent RNA polymerase II to produce messenger RNAs. So here we are making pre-genome RNA, which will go on to be converted to the full-length viral DNA, as well as shorter mRNAs, all of which are used to produce viral proteins.
The pre-genome goes on to make the capsid protein, and from it is also made the P protein, and this is the reverse transcriptase.
So for hepatitis B virus, the reverse transcriptase is called P, not RT, and this is because the protein was discovered and named before it was found to have reverse transcriptase activity.
The P protein is incorporated into the growing capsid, so this now has a pre-genome RNA in it, a full-length
RNA copy of the viral genome. The reverse transcriptase activity of the P protein, which has also been incorporated into the growing capsid, copies that viral mRNA to form first single-stranded minus-sense DNA and then the nearly fully double-stranded DNA. Early in infection, these partially completed capsids containing double-stranded DNA are actually shipped back into the nucleus, where they serve as templates for the synthesis of more mRNAs. Later in infection, these capsids bud into the endoplasmic reticulum, where they acquire a membrane, and then they are released from the cell via the secretory pathway. Let's look at how the process of hepatitis B viral reverse transcription occurs. It begins with a primer, but it's not a tRNA primer as with retroviral reverse transcriptase. It is actually a stem loop primer built into the viral genome, and that's shown here.
It also happens to be a packaging signal. Epsilon means it's a packaging signal. In other words, the capsid protein specifically recognizes this structure, and that's how it's incorporated into new virions. So let's see how this works. This structure is right here. Here's the P protein, the RT,
which binds to the
stem loop and begins to synthesize the complementary strands. It doesn't need a primer like the retroviral RT. It just binds to that stem loop structure and begins to synthesize this sequence, the complementary sequence to this CAUU here. So the enzyme makes some negative strand DNA, and then very shortly after it's synthesized, this negative strand DNA primes in this DR region up at the other end of the RNA, the pre-genome RNA.
So that primer linked to the P protein anneals to the DR1 region, and it begins to prime the synthesis of a DNA copy of the viral RNA, as you can see in step 3.
The RNA template is removed as DNA synthesis proceeds. That's done by an RNase H, which is also part of the P protein. And eventually this copies the entire genome and makes a negative stranded copy. Now it doesn't remove all the RNA, it leaves an RNA primer at the end shown on the lower left. So here's a full-length negative DNA copy. It leaves a little bit of an RNA primer, which you can guess is going to serve to make plus strands. And if you guessed that, you'd be right. But it doesn't make a plus strand going around where you think it would do it. This RNA primer goes up here to the 5' end of the negative strand RNA. So even though it's annealed here, it exchanges this template for this one up here, for the DR2. And there it serves as a primer till it reaches the 5' end. This is very Baroque. It's dead, it can't go any further, but we need to copy the rest. So what's going to happen? Well, this 3' end is complementary to this new product that has been made. And now that will anneal up to the newly synthesized region, and the viral enzyme can complete the synthesis of a plus strand. So this is what ends up getting packaged into virions. So you can see it has some double-stranded character, and in fact, how much double strand depends on the virus preparation. So this reaction making plus strand could go a little bit longer, but it never finishes. So it's partially double-stranded, partially single-stranded. The primer, this RNA primer, the capped RNA primer remains, so that's the piece of RNA that we find in the virus particles. And the P protein, the reverse transcriptase, is left attached to this end of the genome as well. So the 5' end of the minus strand RNA. So that accounts for the weird structure of the viral DNA in the particles. The gap nature, because synthesis isn't finished. The protein, which is the reverse transcriptase, and the RNA primer. So this RNA primer came from here. Here it was left over from the very beginning of reverse transcription. That was the 5' end of the pre-genome RNA. The way that this template exchange occurs is very interesting. If we back up in this process, the RNA primer starts at DR1, but it prefers to go to DR2, and it does so because what we think happens is DR1 folds and makes this stem loop shown on the right here, and it no longer, the 3' end of the primer no longer hybridizes here as a consequence, so it preferentially will hybridize to DR2, and that's what makes the primer transfer to the other strand. So that's reverse transcription of Hep B, and it is significantly different from RT of retroviruses, as you can see. Nevertheless, the proteins are quite similar. So here is a duck hepatitis virus reverse transcriptase. People work on the duck viruses because they have an animal model for infection. They're very similar to the human viruses. And here's the reverse transcriptase of HIV, and these areas of color show where there are amino acid similarities between the two proteins. So you can see there are blocks of similarity in the polymerase region as well as in the RNase H region.
Perhaps both of these proteins arose from a common ancestor.
with an RT. Now, one thing this Hep B does not have is an integrase. So these viruses do not integrate into the host DNA as do the retroviruses.
Of course, they also have a DNA genome, which is different from the retroviral genome.
They have a reverse transcriptase, which is used to make the DNA genome, but they do not integrate.
So let's contrast the life cycles of retroviruses and hepatitis B virus. In the yellow boxes are the nucleic acids that are incorporated into the virions. The top is the replicative cycle of hepatitis B virus, and there a partially double-stranded DNA is incorporated into the virion. That DNA is made completely double-stranded when it infects a cell. It's used by host DNA polymerase to produce mRNAs, the pregenome RNA and other mRNAs. That is copied by reverse transcriptase to form the viral DNA. In contrast, in the retroviral genome is RNA, shown at the bottom. This is converted to a DNA copy by the viral reverse transcriptase. The DNA integrates into the host cell and then is the substrate for the same DNA pol II as Hep B to produce viral mRNAs. There are two other virus families that I'd like to talk about that encode, whose genomes encode reverse transcriptase. And they're similar to the life cycle of hepatitis B virus in that DNA is packaged in the virion. The first example is cauliflower mosaic virus, a virus of plants, as you might guess from the name, cauliflower. It has a DNA genome. As you can see in the yellow box, what is packaged in virions is a double-stranded circular DNA genome. When this viral genome gets into cells, it is repaired and made into a
covalently closed double-stranded circle, same as for Hep B. And then this is the substrate for the production of viral RNA. And the replication or the conversion of this RNA to DNA is accomplished by a viral reverse transcriptase. And you can see there is a primer hybridized to this genome here. Remember, the Hep B primer, there was no primer, there was just this secondary structure in the RNA that the reverse transcriptase used as a primer. So cauliflower mosaic is very much like hepatinovirus. There's one other virus that has a reverse transcriptase, and that is actually another
retrovirus, and these are called foamy retroviruses. They're classified separately from all the other retroviruses because their genome is actually DNA.
Double-stranded DNA. So it's a retrovirus with a DNA genome.
They're called foamy retroviruses because when they were first discovered, the cytoplasm of infected cells was observed to be full of small bubbles, giving the appearance of being foamy.
These encode a reverse transcriptase. The DNA in the virus gets put in the cell. It is integrated into the genome. So there's a similarity with retroviruses. In the genome, mRNA is made, and that is then copied by reverse transcriptase using the primer shown here to make double-stranded DNA that is packaged. So we originally thought that the retrovirus lifestyle was the only way to incorporate reverse transcriptase.
Have an RNA genome from which DNA is made.
And then make RNA again, but it's not true. You can see DNA viruses can have reverse transcriptase.
I want to talk a little bit about retroelements now.
These are retrovirus-related sequences that are present not in viruses, but in genomes of the cell. And there are many of these, as you will see, and when they're integrated into the germline, we call them endogenous proviruses. So in some species, the proviral DNA is actually in germ cells, and it's passed on to subsequent generations that way. Most of the time, these are replication defective. They don't replicate. There are pieces missing, or there are mutations that prevent their replication, although there are a few exceptions which I'll point out. This is the amazing part. Nearly half of our genome is made up of mobile elements, which includes these proviruses and other retroelements. So half of your DNA is related to the stuff we've been talking about today, reverse transcriptase and other parts of tumor viruses. That's a lot of DNA. So here's what these look like. These are the different classes of retroelements which are mainly sequences related to retroviruses. About half of our genome is comprised of these guys. At the top is endogenous retroviruses. So here is a provirus integrated into human or mouse DNA. The purple is the host sequence, and you can see this is repeated. The arrow at these darker purple sections means it's repeated. So that tells you right away that this is probably a retroviral integration site because the host sequence is duplicated. And then we have LTRs and then GAG, PAL, and EMV genes. So you can find these in many species. Humans have between 1 and 100 of these. So they're called human endogenous retroviruses, or HERVs. They have everything that a retrovirus would have, but most of the time they're defective. They have mutations that prevent the formation of particles. So we don't make human endogenous retroviruses because they're all broken. But other species do, like mice have endogenous murine retroviruses in their genome, and they make particles. So the wild mice that you see running around have a few PFU of retroviruses per milliliter of their blood that's produced from these endogenous viruses. And they're passed in the germline in mice. We also have retrotransposons, which are similar to the endogenous retrovirus, but they're usually missing an envelope, so they can't make particles even if they were intact. And yeast there's an example of these in yeast. There are 10 to the 2 to 10 to the 4 copies of these in the yeast genome. Line elements, long interspersed elements, they have homology to retroviruses, but they can't make particles. They have a reverse transcriptase, and they have a poly-A sequence at one end. They have a duplication around them in the genome, which tells you that it's a retroviral integration. And this comprises 20% of our sequence, these line elements. And the best studied one is line 1, which is human, 10,000 to 100,000 copies of these per our genome. And these can jump around, by the way, because if an mRNA is made for this, it encodes reverse transcriptase. If it's active, it can be copied to a DNA form, and it can go somewhere and integrate anywhere else in the genome. So these are mobile genetic elements. If the reverse transcriptase is functional, which it is in many of these, they can move around. So they have implications for diseases of different sorts. They've been traced to a variety of human diseases by interrupting genes. We have short interspersed sequences, the signs, which look like they're about 10 to the 5 to 10 to the 6 in the human genome. There's no reverse transcriptase, and what we think happened here is that an mRNA was made, and then a reverse transcriptase, which was made by a different retroelement in the same cell, copied that to a DNA, and then it integrated somewhere. So the signs in the process pseudogenes, these are probably cellular mRNAs that were copied by reverse transcriptase. So if some of the PAL genes in these retroelements are functional, they can assist in the mobility of genes jumping around. So this is a classification scheme for all the mobile elements related to the ones we're talking about. So here on the bottom you see primates, felines, rodents, ungulates, birds, reptiles, fish, amphibians, and insects. So all these have mobile genetic elements. Yeah.
So those mobile genetic elements, is there cellular integrase that has to integrate them into the DNA? Or do they
it's part of the element. It's part of the element, yeah. So it's part of the PAL region of these elements. Yeah. These just show you how many there are, and many of them are related. We can see here's the HERV species, the human endogenous retroviruses here related to a variety of others from other species. So there are quite a few, and people study these quite extensively to try and understand what they're doing. Not too long ago, a group at Rockefeller took one of these HERVs. There's a family of HERV called HERV-K, which is a fairly recent addition to our genome, less than 1 million years ago. So most of these probably infected us or our ancestors 30 to 40 million years ago. But this is a recent one. So what they did is they sequenced HERV-Ks from lots of genomes and made a consensus that could replicate. They made a DNA that was now functional. They put that in cells and they got particles. So they took a virus which hadn't been around for at least 1 million years, and they corrected the sequence such that when they put the DNA containing that sequence into cells, they got the virus back. So they resurrected a dead virus, essentially. And now they can study this in the laboratory here. So this probably resembles the virus that infected us a million years ago. So this is an example of what you can do. So some of these are very close to being infectious, and you can fix them in such a way that they are infectious. Now, we can watch endogenization in the koala. So go and read this blog post. Koalas have recently been infected with a retrovirus. Koalas are native to Australia, but they have been shipped all over the world. And you can see them being infected in the germline in different parts of Australia. They've also been shipped to Japan. And the ones that go to Japan have the retrovirus in them. So the retrovirus originally infected these animals, and now they're going into the genome. So it's going to be transmitted. And there are groups of koalas in different parts of the country that have this endogenous retrovirus and other groups that don't. And finally, I want to end with this. This is another post you should go read. The fact that our genome contains reverse transcriptase means that other RNA viruses can be integrated into the genome. So Aborna is a negative strand RNA virus like flu, except it has a single strand of negative sense RNA. And this can be found in our genome, Bornavirus DNA. And the idea is it's infected a cell at some point millions of years ago, and endogenous reverse transcriptase from some retro element made a DNA copy of this virus, and it integrated into the genome. So take a look at that. And I want to end up telling you that other forms of life have reverse transcriptase. Myxobacteria, E. coli, even archaea bacteria, reverse transcriptase activity has been found. So this is not something virus specific. So reverse transcriptase evolved long ago before the separation of these archaea prokaryotes and eukaryotes. And we think it might be the bridge between that RNA world that existed first and the modern DNA world. There had to be something to make DNA from that RNA world, and maybe it was reverse transcriptase. So very interesting enzyme.